Welcome to day 2 of our daily revision boosters. Today we're going to be having a look at monoclonal antibodies in our biology blast, alkenes in our chemistry check, and the electromagnetic spectrum in our physics prep. First thing we need to do though is have a look at today's daily formula, which is the one for potential difference. So potential difference is measured in volts, and the way we work that out is the current in amps times the resistance measured in ohms. Today's biology blast is looking at monoclonal antibodies. Now this is only for those of you doing GCSE biology on the higher tier. So if you're doing foundation or combined science, then you can skip on to the next section today. When we're talking about a monoclonal antibody, what we're referring to is something we make in the lab from a special type of cell called a hybridoma. In order to make the hybridoma, what we've done is we've fused or joined a myeloma, which is a cancer cell, with a lymphocyte, a white blood cell. Now the lymphocytes are the white blood cells that make antibodies in our body, and by joining those with our cancer cell, the myeloma, we produce this special cell called a hybridoma. And the hybridomas are the ones that produce monoclonal antibodies. And they're called monoclonal antibodies because they're produced by a single clone of cells. A very important point about these monoclonal antibodies is that they're designed to target a specific type of cell. And the way they do that is by binding to the antigens of our target cell, and then it could actually kill it or prevent it from operating effectively. So just like antibodies in our body when we're talking about the normal action of our white blood cells, then they've got a very specific shape and they bind to an antigen. Exactly the same thing with our monoclonal antibody is that they have a specific shape that will bind to a specific antigen. So when we look at how we make them then, what we start off with is a genetically modified mouse, and we inject them with the required antigen. What then happens is the mouse is going to produce antibodies to that specific antigen, and we then collect the lymphocytes, which are the white blood cells making those antibodies. Those lymphocytes then get fused with the myeloma cells to make that hybridoma. And what we need to do there is fuse them together for the simple fact that a lymphocyte can't exist outside of the body. So what we need to do is fuse them with the myeloma cells so they can survive outside the body, and the myeloma cells also have the advantage of allowing them to reproduce indefinitely. So as those hybridoma cells are reproducing, then they're forming identical clones. So that means that each clone of our hybridoma is going to produce the required antibody, and we can then harvest all of that antibody that's being made. And the antibody that we're harvesting is the monoclonal antibody. So it's specific to that antigen that we originally injected into our GM mouse. So remember those key stages. GM mice are injected with the antigen, the body produces the antibodies to it, we collect the lymphocytes, fuse them with the myeloma cells to make the hybridomas then reproduce, and they produce the monoclonal antibodies which we then harvest. We do need to know then what we use these monoclonal antibodies for, and one of the most common uses is in pregnancy testing. So in those pregnancy test kits that you can buy in supermarkets, Boots, etc., then what you have is a little plastic device which has a little window in it, and then you get a little blue line if you're pregnant. Now the way this works is it's going to be responding to this hormone called HCG, or human chorionic gonadotrophin. And that's produced from about 2 weeks after the date of conception. So what happens is, on that little stick, the little bit that the woman urinates on, then there are antibodies, the monoclonal antibodies to HCG, which are stuck onto little blue beads, as you can see in the diagram on the right there. Now what happens there is, any urine that's got HCG present in it, then the HCG is going to bind onto the antibody on the blue beads, and then as the urine moves up that pad, then it's going to draw those blue beads up with it. Bit further on, you've actually got your window that tells you your results, and you can see that that's got more of these antibodies actually stuck down to the surface in a nice neat line. So that as the urine with HCG gets to that window, what happens again is our monoclonal antibodies are complementary to the shape of the HCG, so they bind to the stuck down antibody, and that means that all of those blue beads then end up in a nice neat line, and you get the blue line saying you're pregnant. The second use of our monoclonal antibodies is in detecting diseases. So what we can actually do is use those monoclonal antibodies to act as a marker, because it will bind to a specific antigen in order to actually confirm that it's present. And a good example of this is in prostate cancer, and it means that we can actually identify the disease nice and early to give an early diagnosis, which leads to better treatment outcomes. The last use that we've got is in treating cancers. Now, monoclonal antibodies can target specific cells and lead to their death or prevent their proper operation, as we said earlier. So what we can do is we can actually design a monoclonal antibody that's going to be specific to the tumour markers that we find on cancer cells. Now, to those monoclonal antibodies, we can then bind our drugs or radioactive substances that are going to have the effect on the cancer cells. And then when we introduce the monoclonal antibodies with the drugs on them, they're going to be attracted to the cancer cells, they bind to the tumour markers on the surface, and will have the effect on those cells, which minimises the damage to the surrounding tissue, thereby reducing some of the side effects of the cancer treatments. Just a few cancers that we've already developed monoclonal antibody treatments for, just in case they ask them. We've got them for breast cancer, stomach cancer and bowel cancer at the moment, and we've got a lot of research going on for other types of cancer which we hope to have in the future. Quick reminder of our daily formula: potential difference in volts is the current in amps times the resistance in ohms. For today's chemistry check, what we're looking at are the alkenes. So this is a concept again only needed in the GCSE chemistry. So if you're doing combined science, just skip on down to the physics section. When we're talking about alkenes, we're talking about another type of hydrocarbon. So they're made of just hydrogen and carbon only. They do form a homologous series, and they've all got the general formula of CnH2n. Now, a key feature about our alkenes is that they are unsaturated. So this tells us that they contain at least one carbon-carbon double bond. Just like with the alkanes, we do need to be able to name the first four straight chain alkenes and draw their displayed formula. So I've given you the displayed formula of one in the bottom right there, which hopefully you know is propene because we've got 3 carbons there. When we come to naming them, then our first 4 alkenes are actually going to be ethene, propene, butene and pentene. There's no such thing as methane because it can't have a carbon-carbon double bond because there's only 1 carbon. So there's no such thing as methane. Your first 4 are ethene, propene, butene and pentene. So remember that. They all end in ene, hence they're alkenes, and the first part of the name is just the number of carbon atoms. Same rules apply for drawing our displayed formula as with the alkanes as well. Put your carbons in a line first of all, put a double bond between 2 of them, and then make sure that every carbon atom has 4 covalent bonds, and then make sure that there's a hydrogen with only 1 bond attached onto all of them. So just go careful with where you've got your double bond not to have an additional hydrogen stuck on there. So remember, the double bond is 2, which means that if you've got the one on the far left, as you can see in the diagram there, 2 from the double bond and then 2 hydrogens, whereas the middle carbon has got 2 from the double bond and then 1 hydrogen because the other bond goes over to the last carbon. So just make sure you use that little check: 4 covalent bonds from each carbon, 1 covalent bond to a hydrogen. When we're thinking about the reactions of the alkenes, then they're going to undergo combustion reactions with exactly the same products as the alkanes, because at the end of the day they're made of exactly the same elements as the alkanes. So complete combustion, which is going to make carbon dioxide and water; incomplete combustion, which is going to make carbon, carbon monoxide and water. The alkenes have something called a functional group. which is an atom, a group of atoms or types of bond in a molecule that are responsible for the characteristic reactions of that substance. In the case of alkenes, it's the carbon double bond carbon functional group. So if they ever ask you what's the functional group in alkenes, then all you need to do is write carbon double bond carbon, just like I have at the top there. The next type of reaction that we find with our alkenes is an addition reaction. Now, as this name suggests, if it's an addition reaction, then we've got atoms or groups of atoms joining together and combining to form a larger molecule with no other products. And a good example of this is the addition polymers, which we're going to look at in one of our future boosters. One of the things that they could ask you to do is to explain how you can identify whether you have an alkane or an alkene. And the test is quite simple. All we do is we take bromine water, we add it to the mixture, give it a little shake, and we look at the colour change. So what we're actually looking for here is the colour change from the orangey brown colour to colourless. If that happens, we have an alkene, because it's undergone an addition reaction, as you can see at the bottom there, to form our dibromoethane. If we don't have that colour change and it stays the orangey brown colour, then it must have been an alkane because it's unable to actually join onto the molecule. Because remember, alkanes are saturated, there are no free bonds available. Alkenes, unsaturated, so the double bond's going to break in order to allow the bromine to join onto the molecule. The last place we can see addition reactions occurring with our alkenes is with hydrogen. So that if we have our alkene, hydrogen and a nickel catalyst present, then we're going to form alkanes. Time for our quick reminder of today's daily formula, which is potential difference in volts is the current in amps times the resistance in ohms. For today's physics prep, we're going to have a look at the electromagnetic spectrum. So the first thing we need to know is the correct order for each of the different parts of the electromagnetic spectrum. So you need to know all their names and the correct sequence that they go in. So radio waves, microwaves, infrared, visible light, ultraviolet, X-rays and gamma rays. Now there are songs that are on YouTube that are actually quite catchy that stick in your head for those, so they're probably worth having a little look at if you struggle to remember them. The other things we need to know are the patterns of the increasing wavelength and increasing frequency. So what we can actually see is that the longest wavelength and lowest frequency are the radio waves, and then you work all the way down the line until you get to the one with the shortest wavelength and the highest frequency, which are the gamma rays. When we're talking about an electromagnetic wave, what we actually have is an oscillating electric and magnetic field. So these two fields are oscillating at 90 degrees to the direction of the wave. So if you have a little look at the diagram at the bottom there, you can see the red ones, those are the electric fields kind of going up and down if you like. And then the blue ones, which are kind of coming out of the screen and into the screen, are the magnetic field because the wave is going kind of from left to right. So you've got your oscillating electric and magnetic fields there, which you can see kind of going in and out, up and down, and they're always at 90 degrees to the direction of that wave. A really good number to remember in our electromagnetic spectrum is 3.0 times 10 to the power 8 meters per second, because that is the actual speed at which all electromagnetic waves will travel through a vacuum. It's also the speed of light, because hopefully we remember that light is just part of the electromagnetic spectrum. So 3.0 times 10 to the power 8 meters per second, remember it. There's two other phrases we should know, sources and absorbers. A source is something that's going to emit electromagnetic waves, whereas absorbers are the ones that absorb electromagnetic waves. And the whole way that we're using these electromagnetic waves then is to transfer energy from the sources to the absorbers. For those of you doing the higher tier, then you do need to know about radio waves. So we need to understand how they're actually going to be transmitted and received. And the way it works is that we've got an oscillating potential difference across a wire making electrons move backwards and forwards. Because remember, if it's an oscillating potential difference, then the electrons will move in that backwards and forwards motion. As a result of that, we're producing a changing electric and magnetic field, because this is happening inside our wire, and that is then being emitted as a radio wave. Those radio waves are able to travel through the air, and then when they meet a piece of metal, i.e. the actual antenna, then the electrons inside that antenna are going to move, and we produce an electrical signal, which is what comes out of your radio at the end of it. The last thing we're going to look at in today's physics prep then are the uses and dangers of this electromagnetic radiation. So first of all, some important uses. If we think about microwaves first of all, then we're using those in mobile phones, satellites, Wi-Fi, all of those rely on microwaves. We use radio waves to communicate in the form of radio and TV. If you want to think about how we can use infrared, then that's in your remote controls. If we're thinking visible light, then that can be communication via ships with obviously sort of flashing lights and so forth, or we could use lasers within our CDs, DVDs, Blu-rays, etc. to read the information off those discs. As we've already said, one of the key things about our electromagnetic waves is that we can use them to transfer energy. So we can actually use them to cook food in two different ways. So the first one is using microwaves. Now hopefully we work out that our microwave ovens will emit a microwave, and those are going to be absorbed by the fat and water in the food. And microwaves are actually capable of penetrating a few centimetres into the food itself. So it's absorbed by the first few centimetres all the way around the food. We then transfer the energy via conduction to the very centre to finish cooking it. The second cooking option we've got is using infrared in our conventional ovens. Now, infrared is only absorbed by the particles on the very outer surface of the food, and then that energy has to be transferred by conduction to cook the rest of the food. So because of this, we can see there's a clear difference between our microwaves that are absorbed by the first few centimetres and the infrared, only the very outer surface particles. So that's why our conventional ovens are much slower at cooking food than microwaves, because it's not penetrating as far in that first instance. There are some other uses of our electromagnetic waves that we should know about as well. Your body actually uses ultraviolet radiation to produce vitamin D, which is why it's really important to actually go outside at this time of year. So if you don't go outside for about 15 minutes a day, then your body's not going to be getting enough ultraviolet radiation in order to actually make the vitamin D it needs. So that means your bones may not be as strong as they should be. We can also use ultraviolet in forensics to identify forged banknotes, identify bodily fluids, etc. And we can also use ultraviolet to sterilize water because it kills any bacteria present within it. If we think about X-rays then, these ones are capable of killing cancer cells. But the more common use you probably know about X-rays is that they produce images of bones, so we can see if something's been broken. The last one we're going to have a look at the use for is gamma rays, which we can use to either kill bacteria or we can use it to kill cancer cells through use of a gamma knife, which you can see in the bottom right there. Even though we've got a lot of uses and positives of electromagnetic radiation, there are also some dangers that we need to be aware of. So the first one is with ultraviolet. So we've said there's a really good use here, that it makes vitamin D in our body, but too much ultraviolet can actually damage the DNA in skin cells and lead to skin cancer. Hence why you should always wear sun creams when you're outside for any extended period of time or in any intense light. The second thing with the exposure to ultraviolet is that if you're exposed to too much, then your eyes can actually develop cataracts. So again, really important to wear proper sunglasses with ultraviolet protection in them when you are spending time in these areas of the world that do have quite intense ultraviolet rays. If we think about our X-rays then, which we've said are really useful to us because we can image bones and we can obviously kill cancer cells, the downside to them is they can also damage cells and cause cancer. So this is why you should not be too exposed to X-rays in your life. So people who are actually taking the x-rays, the radiographers in the hospital, they actually stand behind a LED screen, or they leave the room when they're taking the x-ray to reduce their exposure. They'll also have a badge that they wear that's going to change when the safe exposure limit's been reached, to let them know that they shouldn't be in that room anymore. The gamma rays, they've also got a downside because they can kill or damage cells in the body as well, which hopefully you kind of worked out from the fact that we can use them to kill cancer cells, and therefore if they can kill cancer cells, they can kill other cells too. Hopefully at the end of today's Daily Booster, you've got a better understanding of monoclonal antibodies, you know a little bit more about alkenes and the electromagnetic spectrum. Don't forget to join us again tomorrow for our third Daily Booster in the series.